first presentation is from Oklahoma State University. This is Matthew Couger. His title is Genomic Analysis of the LignoCellulosic Anaerobic Fungus Orpinomyces C1a. So Matthew, go ahead.
So currently a large bottleneck preventing the full utilization of LignoCellulosic ethanol is conversion of non-feedstock plants into constituent monomers that are capable of energy production. The reason this being is that non-feedstock plants are very recalcitrant to enzymatic degradation, and providing more enzymes to allow this process could greatly enhance its ability to become commercially viable. For this analysis, we analyzed the LignoCellulosic fungi, Orpinomyces, a member of the Neocalamastics genus. We did a genomic analysis and transcriptomic analysis of this fungi to identify relevant enzymes that could aid in this conversion process. For our genomic and transcriptomic sequencing, we used Illumina genome sequence by synthesis technology. This is due to a unique facet of this organism having a very high AT content, which makes Roche unable to sequence this organism due to the high amount of homopolymers within the sequence. We generated independent sequencing for both the transcriptome and the genome, and assembled these with two short-read assemblers, Velvet and Trinity. To do these assemblies, we used the NSF shared memory computer Blacklight at Pittsburgh Supercomputing Center. This is the largest coherent memory supercomputer in the world, and it's a very excellent machine that the NSF hosts to do these very large memory jobs. To actually annotate the genome, we took the transcriptome assembly that was produced by Trinity and aligned these transcripts to the genomic assembly by Velvet using a program called PASA. This allowed us to delineate the exon-intron structures of these genes and to fully annotate the organism. Using this information, we trained eukaryotic genome callers and created consensus models using EVM to create the final models for the organism. Functionally relevant genes were categorized by using a combination of homology search with NCBI BLAST, and PFAM annotation. So if we look at the genome came out, we have around 100 megabases of assembly from the genome. This was from 146 million paired-in reads from the Illumina. We have an N50 of around 1,600 base pairs with the N90 of 520. The AT content range is anywhere from 84 to 94% on the contigs, with an average AT content of 87% for each contig. For the transcriptome, we had about 176 million read pairs, which resulted in around 25,935 transcript models that were greater than 300 base pairs. The average length of a BLAST-X transcript that has an e-value of either -5 or less was 1,075 base pairs. The total number of consensus models that we got from annotating the genome was 14,564, with the average length of each model with the top hit of either -5 or less being 350 amino acids. Through PFAM domain identification, we classified which ones would be functionally relevant by homology to members of the CASI database. We came up with 392 models that potentially have a GH domain of either -4 or less, 588 models with the CBM domain, 83 models with the carboxylic esterase domain, 40 models with pectin metabolism, 59 with polysaccharide deacetylase, and 65 with sugar and other transporters. Classification of these GH domains into families shows that this genome has expansion of 3 main categories, mainly with endoxylenase, hemicellulase degradation, ferryl esterase degradation, and expansion of cellulase K-like cellulases. And with that, I'd like to thank EPSCoR for funding the research and the NSF for the supercomputing capabilities to allow this research to happen. Thank you.
Thank you.